One of the most important functions of the cell membrane is the ability for it to bring things into the cell and to allow things to leave the cell. If you remember back to selectively permeable or semi-permeable, that just means that it can bring things in or out, but it gets some control over that. So not everything gets to come in or out. So when something is moved into the cell or out of the cell, it is done either passively, meaning it takes no energy, and we're going to go down a gradient. Or it's done actively, which means it has energy, and we're going to go up a gradient. So we'll talk about passive first. The first type of passive transport is called filtration, and this is when pressure forces fluid through a membrane. So in your blood, you have a high pressure system, and outside of the blood, it's lower. So as your blood moves through these porous capillaries, water is forced out of the blood into the fluids surrounding your cells. The second type of passive transport is called diffusion. Diffusion is the net movement of particles. So now we're not talking about water anymore, we're talking about particles down a concentration gradient. Okay, so here we've got a picture. We've got this glass. There's some kind of membrane here in the center. The membrane is porous, and these red molecules are able to fit through this membrane. There is a concentration gradient over here. We have a high concentration of red dots on the left. We have a low concentration of red dots on the right. These little brackets just mean concentration of. So, without needing energy, these red particles are going to want to spread apart. When they're really close together like this, they bump into each other. And when they do that, they speed away from each other. Imagine being stuck in an elevator, and you're going to kind of move apart from the other molecules or other people in the elevator. So over time, most of these red dots over here are going to come over to the right. Eventually, you end up with two sides where the concentration of red dots is relatively equal. Not every red dot moves in the same direction. These are moving randomly. So this red dot might have actually moved to the left, but since so many more are moving over to the right, we end up with an equilibrium. So that's what I mean by net. Not every single particle moves down a concentration gradient, but overall, most of them do. We can make diffusion go faster by increasing the temperature, by increasing the steepness of the gradient, by making it more surface area, and by making it more permeable. The molecular weight of the molecule will also affect how fast diffusion goes. The bigger this red dot, the slower it's going to move across. Really small red dots are going to be able to move through easier. So what happens when the red dot is no longer actually able to move through the membrane? We get something called osmosis. So osmosis is, again, net flow of water across a semi-permeable membrane. So water's special, it gets its own name here. Osmosis happens when the molecule cannot pass through the membrane, right? Okay, so here we've got a beaker. It's got a membrane here. Side A, we've got just water. These little blue dots, those are just the water molecules. Side B, we've got some larger molecule here. Maybe it's glucose, a protein, something. And there's water in here too. This membrane, the holes in this membrane are too small, and this big molecule can't pass through here. But it still wants to be separated. There's still pressure that these molecules want to be as spread out apart as they can be. So that is called osmotic pressure. And that draws the water in to help push these molecules apart from each other. So the more concentration difference I have on the right, the harder it's going to pull that water in. So over time, what's going to happen is the water is actually going to come into this side. And instead of talking about the concentration of whatever this yellow thing is, we're now talking about the concentration of water. So if you think about this, here's a kind of a classic example. We have a tube. Let's put a membrane in the center. There's an equal water level to start, and we've got these red dots on one side and maybe a couple on the other. If these red dots could move through the membrane, they would move from left to right to make an equilibrium, and that would be diffusion. But my membrane is not permeable to red dots. It is only permeable to water. So what's going to happen is the water is going to move from an area of high concentration of water, right? There's more space over here, that's the water, to an area of low concentration of water, right? Over here, because we have more red dots, we have less water on this side, so the water is going to move that way. So what's eventually going to happen is this water level is going to come down, and this water level is going to go up. So we use this osmotic pressure to regulate the flow of water into and out of our cells. All right, so that's tonicity, is the ability of a cell to affect its fluid volume. And we want to kind of maintain the same fluid volume all of the time. So here's a red blood cell in the bottom in a hypo, iso, and hypertonic situations. So let's start with iso. Iso means same. So in an isotonic solution, here is a little beaker. Let's call this iso. Here's my red blood cell. We've got about the same concentration of solutes inside the cell as we do outside the cell, right? So there's not really a difference between the inside of the cell and the outside of the cell. This is what we like for our blood, right? We want the inside of our blood cells and the outside of our blood cells, our blood plasma, to be about the same concentration. So some water randomly moves in and some water randomly moves out. Again, this is a passive transport at about the same rate. So there's really no change. Blood still stays the same size. All right, let's look at our example in a hypotonic solution. So here's my same beaker, here's my same blood cell. Hypotonic, my cell's gonna have stuff in it. Hypotonic might be something like distilled water. And in this situation, the cell has a higher concentration of solutes than the surrounding water. All right, so what's gonna happen here is the water wants to go from an area of high concentration, which is outside the cell, to an area of low concentration, which is inside the cell. So we're going to see a lot of water move into the cell and very teeny amounts of water moving out of the cell. So what's going to end up happening is the cell is going to get bigger. And in human, we don't have cell walls. The cell could actually burst or lyse, right, to break apart. So hypo means less. The concentration of solutes is lower. Hypertonic, we've got our cell. It's got stuff in it, but it's in a very highly salty or really sugary solution that's got a lot of solutes. This takes a minute to draw all these dots, right? And so the water it's in, this would be really salty. So this is why if you're stranded in the ocean, you're not supposed to drink the salt water, because it will actually make your blood very salty. So in a hypertonic solution, you can see more white space inside of the cell. So there's more water inside of the cell than relatively outside of the cell. So you're going to see the water move out of the cell. Very little water might move in. And what ends up happening is this shell is actually going to crenate or shrivel up. Look at the structure of these red blood cells, right? This big one, and then this shriveled one. Are those going to function the way you want a white blood cell to function? Probably not. Things can also move through diffusion. So this is still diffusion. This is still passive. But they have some help, right? If you're facilitating someone, you're helping them. So in this case, we're going to have molecules still going down a gradient, right? From high concentration to low concentration. But now they're going to pass through some sort of channel protein that's going to help them get across. This might be because this molecule is extremely polar, meaning it can't get through this hydrophobic region very well, or because it's really big and it just needs a protein to help it get through the membrane. All right, active transport is going to require energy, and we're going to go from low concentrations up a gradient to a high concentration. There's a couple of different types. Primary is what we're going to focus on. The best example of primary is the sodium-potassium pump, which is pictured right here. So what this does is it moves 3 sodium ions out of the cell, and it moves 2 potassium ions into the cell. Notice that we're going from few potassiums to many potassiums. That's going against a gradient. We're going from few sodiums to many sodiums. That's going against the gradient. This requires, there it is, ATP. Secondary active transport is when we have active transport in one location on the cell, and we use energy to move, in this case, sodium across. And that's going to help power passive transport in a different location, right? So if we didn't have this pump over here moving the sodium out, we would end up with a buildup here, and the sodium wouldn't be able to get in on this side. So to keep sodium coming in on this side passively, we have an active pump set up over here to help kind of move it through the cell. The last type of active transport is called vesicular transport. So this is also active. And this is going to be how we move really, really big things. These are going to be moved in what's called a vesicle. So this is a vesicle, is a little pocket of cell membrane inside of the cell. Exocytosis, let's start with exocytosis. Exo means out, right? The outside. So exocytosis is how we move things from the inside to the outside of the cell. This is how we secrete proteins out into the fluid. Endo means in, right? So endocytosis is going to be how we move things in. Phagocytosis is kind of like cellular eating. So we get large solid particles. Looks like a bacterium, gets swallowed up by this white blood cell and put inside of a vesicle. Pinocytosis is kind of like drinking. I remember this one because Pinot is a wine and you drink wine. So this is going to create a vesicle by kind of invaginating in and just drawing in any fluid right next to it, and then eventually making a vesicle inside of the cell. Receptor-mediated endocytosis. This is kind of similar to pinocytosis. It's going to pull things into the cell, but it's got receptors right here. This one is specifically looking for stars. The pinocytosis, it just pulls in whatever's next to it. It doesn't get to choose between these blue squares and these yellow stars. The receptor-mediated is specifically choosing the yellow stars to bring in that molecule. Take a moment and contrast between passive and active transport, and what are the differences between these two processes.